Good morning, students. Now in the today's period, we can study here the Operon concept. What is the Operon concept? It is a transcriptional mechanism of gene control. We know that there is transcription, that is the DNA language is transferred. So it is a transcription mechanism of the gene control. In 1961, Jacob explained this metabolic pathway. So a scientist who worked there, that is Jacques, in 1961 explained this metabolic pathway. And in the Escherichia coli, E. coli, in the Escherichia coli, when lactose is present, we know that there are different types of sugars. This is lactose, sucrose, glucose, and all this type. If the lactose is present, आपल्याला माहिती आहे की दूध हे गोड लागतं, त्याच्यामध्ये लॅक्टोज नावाची शुगर असते. कधी कधी लहान बाळ त्याच्या आईचं दूध पचवू शकत नाही, त्यावेळेस आपल्याला काही वेगळ्या मेकॅनिझमच्याद्वारे त्याला ते डायजेशन वाढवावं लागतं. सो दॅट इज ऑल्सो कॉजेस वन डिसीज. पण या ठिकाणी आपण आपल्या अभ्यासामध्ये इश्चरिशिया कोलाई इफ प्रेझेंट with lactose sugar, there are three different types of enzymes are necessary. Which are these three different types of enzymes? These are beta galactosidase, second is beta galactoside permease and trans-acetylase. So three types of enzymes are necessary if the lactose is present in the E. coli or the medium of the E. coli. So what is the functioning of these three different types of enzymes? Beta galactosidase, it digests lactose to glucose and galactose. What is the function of the beta galactosidase? It digests the lactose to glucose and galactose. या ठिकाणी लॅक्टोज शुगरचं रूपांतर ग्लुकोज आणि गॅलॅक्टोजमध्ये करण्याच्या कामी हा बीटा गॅलॅक्टोसाइडेज हा एन्झाईम येतो. सेकंड, बीटा गॅलॅक्टोसाइड परमिएज. बीटा गॅलॅक्टोसाइड परमिएज, इट परमिट्स द लॅक्टोज मॉलिक्यूल टू एंटर इन द सेल. इट परमिट्स द लॅक्टोज मॉलिक्यूल टू एंटर इन द सेल. थर्ड एन्झाईम ट्रान्सॲसिटायलेज, it transfers an acetyl coenzyme A to galactosidase. These are different types of functions of these enzymes. So if the lactose is present, three different types of enzymes they are necessary, and these are beta galactosidase, then beta galactoside permease and trans-acetylase. And the synthesis of these enzymes are controlled by DNA segment. It is called as Operon. So these three different types of enzymes they are necessary if the lactose sugar is present, and here a small DNA segment, that is Operon, is responsible, or it is useful to control the production or synthesis of these different enzymes. So we know that here operator side O and three different structural genes, that is ZYA. ZYA are necessary. The action of structural gene is regulated by the operator site. The action of structural gene is regulated by the operator site with the help of repressor protein, with the help of gene I. What is I? Small I. That is inhibitor, and it is called as the regulator. What is the regulator gene? Here the action of structural gene is regulated by operator site with the help of repressor protein, and a small gene, that is small I, that is inhibitor, that is called as regulator gene. If operator is switched on, we have to learn here two processes. Operator is switched on, three genes, that is Z, Y and A, are transcribed by RNA. RNA transcribed Z, Y and A, these three different types of genes, and the polymerase, that is RNA polymerase, into into single mRNA. So ZY and A are transcribed by RNA polymerase and into the single mRNA. Each structural gene is called as cistron. We know that there are different names: cistron, racon, muton. So here each structural gene is called as cistron and it transcribes long mRNA covering various cistron. Covering the various cistron, that is the long mRNA covering the various cistron, it is called as polycistron. So keep some words in your mind. If operator is switched on, three genes Z, Y and A are transcribed by mRNA polymerase into single RNA, into single mRNA, and each structural gene is called as cistron. Now here each structural gene is called as cistron and it transcribes the long mRNA which is covering the various cistron called as polycistronic condition. Polycistronic condition. If operator is switched off, by a protein called as repressor, when this protein is attached to operator and block the switch off, and structural genes are not expressed. What is the condition when the operator is switched on? Three genes Z, Y and A are transcribed by RNA polymerase into single mRNA and each structural gene is called as a cistron, where the transcription of long mRNA which is covering the various cistron is called as polycistronic condition. If the operator is switched off by a protein called as repressor, when this protein is attached to operator and block it, the protein is attached to the operator and block it, and it's switched off and structural genes are not expressed. This is generally theme of the lac operon. Now, what is the lac operon? Lactose of E. coli is inducible operon. Lactose of Escherichia coli is inducible operon. The operon is switched on if chemical inducer lactose is present in the medium. So we know that here lactose is the important factor in the medium. So lac operon has three steps. What is operon? Synthesis of different types of enzyme, beta-galactoside permease, then the beta-galactoside sidase, then the trans-acetylase. They are synthesized and they are controlled by the DNA segment called as operon. And here lac operon, when the lactose of E. coli, Escherichia coli, is indivisible operon, the operon is switched if the chemical inducer lactose is present. So it has different steps. त्याच्या वेगळ्या steps आहेत. One is regulator gene. Lac operon has 5 steps. One is regulator gene. It controls operator gene. Regulator gene, promoter gene, operator gene, structural gene, and inducer. They are present here. So here in the lac operon model, the regulator gene is present. It controls operator gene and with inducer present in the cytoplasm. Regulator gene precedes promoter gene. Regulator gene precedes promoter gene. Repressor binds with and stops its action. So how the action stops, that is we have to study in the lac operon model. So second gene is called as promoter gene. It precedes operating gene or operator gene. It marks the site where RNA polymerase enzyme binds promoter gene, that is the best sequence, and strand act as a template. So what is the template? Template is able to produce or able to give its genetical language. So here promoter gene is present. Regulator gene, it controls the operator gene with inducer present in the cytoplasm. Regulator gene precedes promoter gene. Repressor binds with and stop this action. Promoter gene, it precedes operator gene it makes or marks the site where RNA polymerase enzyme binds the promoter gene base sequence and stand act as a template. Now third one is operator gene. It controls the functioning of structural gene. It controls the functioning of structural gene. What is the structural gene? When lactose is added in E. coli, the gene produces mRNA which produces different types of LAC-Z, LAC-Y and LAC-A types of structures on the ribosome, and these enzymes are produced, that is the beta galactosidase, then beta galactoside permease and trans acetylase. They are produced by the LAC-Z, LAC-Y and LAC-A that is present on the ribosome. Here the three different types of enzymes, they are necessary as we have studied previously. So here the production of enzymes takes place, and now the inducer. It is chemical present in the cytoplasm called as allolactose, which inactivates the repressor. Allolactose, it inactivates the repressor when lac operon is switched on. Here inducer, it is a chemical present in the cytoplasm which inactivates the repressor when lac operon is switched on. Then the inducer joins repressor protein. Inducer joins to repressor protein, preventing the binding of repressor to operator gene. What it prevents? The binding of repressor to operator gene. So operator gene is free and RNA polymerase can move from promoter to structural gene via operator gene. So that is the 3 or 5 different steps which are necessary or useful in the lac operon model. Here lactose is present, 3 different types of enzymes they are necessary. They are produced by the LAC-Z, LAC-Y and LAC-A. Here the 5 steps are present there, that is the regulator gene, promoter gene, operator gene, structural gene and the inducer. So this is related with the lac operon. Thank you.